The Cell Cycle by Mario Gonzalez and Amy Gonzalez.
What is the cell cycle? The cell cycle occurs throughout the entire human body. In order for the cell to replicate itself, it has to go through interphase and mitosis. The interphase portion of the cycle occurs first. There are 3 stages involved in interphase. They are called the G1 phase, S phase, and the G2 phase. After these stages have passed, the cell moves on to mitosis. During the process of mitosis, 4 stages occur. The stages include prophase, metaphase, anaphase, and telophase. After the cell undergoes all of these stages, 2 new daughter cells are produced. In the diagram in the upper right corner, it shows all of the stages of the cell cycle.
Why does the cell cycle occur? The cell cycle occurs because it plays a crucial role in survival. Through the use of cells dividing, our bodies are able to maintain homeostasis. Homeostasis is the human body's ability to maintain a balance and stability in an environment. Examples of the cell cycle maintaining equilibrium in the body can be seen when keratinocytes in the stratum basal and stratum spinosum layers of the epidermis divide. They are produced in order to replace dead keratinocytes on the superficial layers of the epidermis. It is also seen when injuries occur in the body, whether it be a cut, nerve damage, or a broken bone. Cells in the affected area divide and multiply in order to repair the damaged areas. As seen in the diagram, the keratinocytes newly formed in the spinosum or stratum basal area of the epidermis gradually move up the layers.
Interphase. Interphase is the period of the cycle that has to deal with the cell growing, preparing for its division. This is also the longest period of the cycle. As stated earlier, there are 3 subphases that take place during interphase. These are the G1 phase, S phase, and G2 phase. Each one of the subphases has a crucial role in the growth and preparation of the cycle. The white arrow pointed in the diagram shows these phases in interphase.
The G1 phase. The G1 phase, also known as the first gap phase, is known for cell growth. As the cell grows, it continues to develop and carry out its normal functions that are needed for a specific cell type. As a part of the cell growth, protein synthesis occurs at a very fast rate in order to create organelles and new components for the cytoskeleton. Following this, the cell continues into the S phase unless the cell is amitotic. If the cell is amitotic, the cell will never divide and remain in the G1 phase, known as the G0 phase or the G0 phase. In the diagram, it is seen all the components needed in order for the cell to grow.
S phase. During this part of interphase, this is where the DNA is replicated or synthesized, hence the S in the name. During this replication process, the chromatid unravels and a new DNA strand is formed by duplicating the base pairs of the original strand of DNA. This is done by using the original DNA strand as a template to build a new one. The diagram in the upper right corner shows all the stages needed for DNA replication.
Steps in DNA synthesis. In the diagram, you see the first 2 steps in order for the DNA to replicate. First, an enzyme called helicase comes in and unwinds and separates the original DNA into 2 strands. Secondly, an enzyme called primase attaches itself to a strand of the original DNA in order to form an RNA primer. Primase attaches itself to both strands individually. The reason an RNA primer has to be formed is due to the fact DNA polymers cannot build a new strand of DNA from scratch. Having an RNA primer gives it a starting point or a reference. A good way to remember this is thinking of paint. In order to begin painting a wall a different color, you must put a primer on the wall as a starting point.
In the picture in the diagram, it is the last two stages of DNA synthesis. And in the third phase, DNA polymerase attaches to each strand of the original DNA, and the new DNA are strands finally being built. The way this happens includes the polymerase starting at the RNA primer and building on from there. The polymerase attaches free nucleotides to the template made from the original strand of DNA in accordance to the rules of complementary base pairing. Note, during this process, on each strand of the original DNA, the new strand of DNA is being built in the opposite direction of each other. Lastly, the DNA synthesis is complete with a product of two identical strands of DNA, creating two new double helices.
The G2 phase. The G2 phase, also known as the second gap phase, is an additional growth phase. In this phase, the centrioles are duplicated from the aid of rapid protein synthesis necessary for cell division. By additional cell growth and protein synthesis, the cell uses this phase as preparation for division, known as the M phase or mitosis.
M phase. Once interphase has been completed, the cell division is ready to start. This is known as mitosis, hence the M in the name, where the replicated DNA divides into two daughter cells. This process happens through four phases called prophase, metaphase, anaphase, and telophase. During this phase, the cell also undergoes a process called cytokinesis, where the cytoplasm is split. At the beginning of the M phase, the cell contains 92 chromatids before this complex set of events takes place.
Prophase. During this process, the nucleolus begins to become compact and a mitotic spindle fiber forms. The centrioles move to the opposite poles of the cell, and their spindle fibers attach themselves to the centromere, which is the center of the sister chromatids. Concluding the prophase, the nuclear envelope breaks apart completely. As shown in the diagram, you can see the nuclear envelope degenerating through the dashed lines, and the centrioles are coming into the cell as seen on the left side of the cell.
Metaphase. This period of mitosis is the longest of all the four phases. Here is where we see the sister chromatids align in the center of the cell. This is able to happen because the spindle fibers of the centrioles pull back and forth until the sister chromatids perfectly line up in the middle. A good way to remember this is by looking at the first letter of the metaphase. The M can be used to remember the chromatids line up in the middle. And in the diagram, it is shown that the chromatids align at the horizon.
Anaphase. The sister chromatids separate and are pulled towards each pole of the cell in this phase of mitosis. By the spindle fibers shortening, the cell causes these chromatids to gravitate toward each pole. Occurring simultaneously, the spindle fibers not attached to any sister chromatids get longer as opposed to getting shorter. This causes the cell to elongate along with the process of cytokinesis beginning. Like metaphase, a good way to remember this is also looking at the first letter of anaphase. The A can be used to stand for away. Shown in the diagram, you can see the beginning of the sister chromatids being pulled towards each pole of the cell.
Telophase. This is the final step of mitosis and the cell cycle. Here is where the daughter cells separate. Nuclear envelopes form again, and the DNA takes on a looser build of chromatin, making the chromosomes less visible. At the same time, cytokinesis is occurring. This is where the process of the cytosol and its organelles are distributed evenly between the two new daughter cells. This forms a cleavage furrow, and this is where the cells split apart.
Final product. Resulting from this process is two cells that are identical that will now continue the cell cycle going into interphase.